Ecology is the study of the relationships between organisms and each other, and between organisms and their environment. In this study, we examine the relationship between trees, herbivores, and fungi. Trees can be infected with fungi, which are organisms that feed on organic matter, such as molds and mushrooms. They can be infected with them at their roots, along their trunk and branches, and all along their leaves. In some cases, this can leave the tree quite damaged. Another factor that impacts tree health is herbivory. Herbivory pertains to how much of a plant is eaten by herbivores, which are organisms that are primary consumers in terms of trophic level, because they get their energy from consuming primary producers, which are autotrophs and plants, which make energy directly from the sun by way of photosynthesis. Our purpose for our research was to answer the questions: what kind of relationship do herbivory and fungal damage have? Do herbivores prefer less damaged leaves when they choose where to have their next meal? Each of us got 10 leaf samples from 2 different trees to analyze the amount of fungal damage and herbivory with relation to the leaf's surface area. Most of us ended up picking one native and one non-native tree. Native trees are trees that are naturally in the ecosystem and that they are present in. In this case, that would be in Raleigh and the surrounding area. Non-native trees, on the other hand, are the opposite and are not naturally found in this area. They had to be brought over from another place. This may sound unimportant or unrelated to our research, but whether a tree is native or not can impact its resilience and survivorship. In this case, it could possibly affect how fungi and herbivores are able to interact with trees. We hypothesized that herbivores tend to eat leaves without fungal damage, meaning that if a leaf was damaged by fungi, herbivory would not be seen as much in that leaf, and that it would not make a difference whether the trees that the leaves came from were native or not.
So for our data collection, each group member did a random sampling on 2 trees, and we each took 10 leaves per tree, measuring their surface area of each leaf in square centimeters. We randomly generated a sequence of numbers in order to select the leaves, and we took a direction to select the branch, and then a number for each position up the leaf. We then measured with a clear plastic with a grid of square centimeters on it and documented what the tree species was and the location. We collected this into an Excel spreadsheet, and then we ran metadata on it, and a meta-analysis, in order to identify a hypothesis. We ran a lot of analysis on the trees. We looked at whether they were native or non-native, what time they bloomed, what time their leaves fell, their taxonomic data, whether they were hardwood or softwood. We compared the surface area damage, their herbivory, their fungal damage, the ratio between those two, and more. The results of our hypothesis were correct. We found that the leaves with more fungal damage had less herbivory damage, and less fungal damaged leaves had more herbivory damage. So on our first graph, you can see that. On the left, in the green, is leaves with more herbivory damage. And you can see they tend to have less fungal damage. And on the right, we have trees with more fungal damage. And these really do have less fungal damage. Our outlier in this case is the ginkgo tree. It did not have any damage, or it had very little damage, and so it did not show up here. Our next graph didn't show up the first time, but it showed up the second time. So here it needs a little bit more explanation. This is average percent damage graphed against the absolute value of the difference between herbivory and fungal. So what this means is that the blue bar represents the total damage, and the red bar represents basically the maximum or the amount that the larger of the two took. So when this bar is very close to the maximum, like over here, here, and here, this means that a large percent of the damage, was just one type of damage, which is consistent with our hypothesis. This would be an outlier in our group here, according to this graph. As you can see, the red bar is not close to this blue bar, meaning that multiple types of damage were accounted for on this tree.
So for the reflection, like we stated earlier, the study of ecology dives into the relationships between organisms and their environment. The study focused on the interconnection that trees, herbivores, and fungi have with each other. The point was to explore the dynamics between herbivory and fungal damage on tree health, while considering whether herbivores exhibit a preference for leaves with less fungal damage. So throughout the data collection experiences that each of us had, we each had to take some specific factors such as bloom time, leaf fall time, taxonomic data, and the nature of the wood itself, as in whether it's softwood or hardwood. The percentages of surface area damage, herbivory damage, and fungal damage were also looked at, as well as the ratio of fungal to herbivory damage. So by the end, our combined efforts ended up supporting our initial hypothesis, which is leaves with more fungal damage experience less herbivory damage, while those with less fungal damage experience higher levels of herbivory. So pretty much this correlation suggests a relationship between herbivores and fungi, where the presence of one may influence the activity or presence of another.
Based on our results, it can be concluded that leaves that contain higher amounts of fungal damage tend to have reduced levels of herbivory damage. There are numerous limitations to this test, however, as only 8 different species of trees were tested, and each tree was only tested once. There is a very realistic possibility that you may receive differing results if the experiment was done again. That's because maybe you have a species of tree that does not apply to these rules, and maybe you have loads of herbivory damage and loads of fungal damage on one tree. Or maybe you tested the same tree again, like the ones we tested, and it doesn't match up. You get the same thing, you get loads of herbivory damage and loads of fungal damage. Because for the 8 trees that we tested, we got 8 trees that had either loads of fungal damage and no herbivory damage, or loads of herbivory damage and no fungal damage. So maybe the data doesn't match up there. But with that said, the 8 different species of trees were tested, and every single tree matched our hypothesis. And we didn't do any cherry picking. We didn't do any of that. So with even one of the trees for each species being tested, the fact that every single one of those trees matched up adds a large degree of validity to our experiment, because what are the chances to get 8 random trees of different species and they all have the same data? They all match exactly the same. With that said, though, it'd still be wise to do another run through the experiment just to make sure and hammer home the point and validate our results. So now what does this mean? What can we do with this? So given what results the experiment provided, we can use this information to make a broader and more general conclusion. Theoretically, based on these results, regions with large amounts of fungal activity and fungal damage are going to be far less likely to have large amounts of herbivores when compared to an area with reduced fungal populations, because the herbivores are not going to want to be in an area where every single tree has 37 mushrooms growing on it and everything's rotting away and getting just destroyed by the mushrooms. So you could use this, scientists could use this, or ecologists or whatever could use this idea to predict where there might be a lot of herbivores. So if there's like an area of loads of fungal damage, you can predict that, okay, maybe there's not going to be that many herbivores there. Or on the contrary, if there's like no fungal damage, there's going to be a lot of herbivores there. So you can kind of predict via that what you might expect. Because according to our data, they directly contradict one another. Again, if you have more fungal damage, you're going to have less herbivore damage in contrast. So